No para mí, ¿no?
No para ella. Sí.
No, sí, otro lado.
The presentation will you have to go on.
No. I'm used to that. Are we good to start, or I don't know? Okay.
Yes. Yes. Okay. Good morning. Thank you for coming here. This is the first round of the presentation of the Professor Phillips. Professor Nicole Phillips is come from the University of Southern Mississippi. She have a lot of project and publication in conservation, but the principal focus of she is conservation with focus in genetic of genomics. So today is a seminar from the graduate student, and the idea is just to present this title. So enjoy.
Thank you so much. And thank you to everyone who's listening online or remotely. I'm really happy to be here and to share the different kinds of research that I do in my lab in the United States. Today, I'm focusing on conservation genetics. And Tuesday and Wednesday, I'll be talking about what's called environmental DNA. And don't worry, if you don't know anything about those topics, the point is to teach you and explain what this research is and means. So I want to start basic because I know everyone comes from different backgrounds, so that this is understandable, hopefully, to everybody. So the field of conservation genetics, what we do is we use molecular data as a tool. So I don't study DNA or molecules for the sake of studying those DNA or molecules. I use molecular tools to better understand the ecology of threatened species. And so the goal in any type of conservation genetic study is really to get data that's going to help understand species to reduce the risk of extinction. That's what we're trying to do. We want to prevent extinctions of species. And this is a very applied field. The results are always, we hope, used by wildlife managers and conservationists to try to protect species better. I have studied conservation genetics for almost 20 years now, and I've worked on a lot of different taxa. I've worked on sharks and rays, I've worked on marine bony fish, freshwater fish, even marine mammals, and I had a very, very brief 6-month project on invertebrates a long time ago. But today, I'm going to talk about conservation genetics of sharks and rays in particular, because they're my favorite group. Why? Why do we care about sharks and rays? They're extremely important. They are economically important. So there are shark fisheries around the world that many people rely on, especially island nations. They rely on shark meat for protein. And of course, economically, tourists, people like to get into cages with white sharks. I know I do. Maybe that doesn't sound fun to you, but there's a lot of economic, this is a big economic driver in a lot of places. They're also culturally important to many different people and groups around the world, and ecologically important. So as someone who's a molecular ecologist or, conservation geneticist, ecologist, this is what I really care about a lot. So sharks and rays are what we call mesopredators. That means, or apex predators. That means that they are mid-level to high-level predators in ecosystems. So they have the ability to influence the abundance of fish, the behavior of fish. If you have healthy predator populations, you can reduce the likelihood of invasive species. So those are species from another location coming in and sort of ruining the ecology of the ecosystem. And in this way, they also prevent transmission of disease by eating the sick fish. And so sharks and rays actually have the ability to support ecosystem stability and reduce impacts of climate change just based on the role that they're filling in the ecosystem. Again, why sharks? This is the IUCN Red List. I'm going to explain this because I'm going to keep coming back to this. I'm going to come back to this on Wednesday as well. If we look here, it says more than 42,000 species are threatened with extinction. This is a lot of species. And when people think about species on the verge of extinction, you usually think about tigers or snow leopards, terrestrial mammals. Well, the mammals, only 27% of mammal species are actually threatened with extinction. If you look at this, the sharks and rays, 37%. So that's among the most threatened groups of all taxa. Now, what the IUCN Red List does is it looks at what is the risk of this species going extinct. And it does this in a quantitative way. That way it's the same for each species. These are what we call the categories. So scientists get all the data we have and they make an assessment of what is the likelihood this is going to go extinct. These in red are what we call the threatened categories. So critically endangered, this is the highest level. If things get worse, the next category is extinct in the wild. So species that are critically endangered are in a lot of trouble. And I typically study species in this ranking of critically endangered. So it is we call it a crisis discipline, where we're trying to do anything we can and gather any data we can to prevent extinction. And so if you look at this, these are all the sharks and rays. There's just over 1,200 species. And a decent percent of them are critically endangered. It's about 36%. We also have, threatened, so all of these categories. 3 might already be extinct. We don't have enough data on them. We have species that are what's called near threatened. And what that means is if the situation gets worse, they're going to then be at risk, some risk of extinction in the wild. Least concern, these are the ones that we're not worried about them. They're okay. At least for the next 3 generations, they should be okay. And then we have this other category, data deficient. This means we don't even know. It could be about to go extinct. It could be fine. But we don't even have enough data to say or assess that extinction risk. So I'm going to keep coming back to this throughout this seminar. But what you'll find is that we're really talking a lot about, well, species in these categories overall. So they're at some level of risk of extinction, but especially this one where the situation is dire. So why are they in this situation? Why are so many sharks and rays at risk of extinction? They are being exploited or directly targeted for their fins and for their meat. Meat is mostly secondary. It's mostly the fins that they're being harvested for. So a lot of these fins are used in soups. They're considered a delicacy. It is considered a status symbol of high class if you can afford to eat shark fin soup in some countries around the world. But they're also being caught as bycatch, and this should not be underestimated. So bycatch is where fisheries are trying to catch maybe some species of bony fish, tuna. And they catch tuna, but they are also catching a lot of other species that they aren't interested in. And sharks and rays, this is a big problem. They're accidentally catching these species, and so we have mortality through this bycatch as well. And this bycatch is a driver of the threatened status for many, many species. Then we look at what we call their life history or their biology. Most sharks and rays are long-lived. How long? For most species, we don't know. Greenland sharks, which are pictured here, these are estimated to live up to 400 years, so extremely old. But for most species, we really have no idea how long they even live. They tend to sexually mature later in life. You're going to see this is a trend for most species. We don't know how old. But when I say late in life, this might mean they start reproducing at 8 years old or 15 years old, as opposed to a little fish, which might reproduce in year 2 or 3. And they also have low fecundity. So this means that their reproductive rate is very slow. So again, while bony fish usually have mass spawning of eggs and sperm in the water, potential for many, many young in any event, most sharks and rays have internal gestation. So the development of young happens inside, sometimes in eggs, but my poster child for low population growth rates, this is a cownose ray. This is a species that I work on in my lab. They have 1 pup every other year, 1. And the chance of that 1 surviving long enough to reproduce itself is very low. So because of this, we say they are susceptible to overexploitation. Well, what that means is that the rate at which animals are being taken from the population is greater than the rate that the population can increase. And so because of that, we see population declines. So this is why I study this group, because they're in a lot of trouble and we know very little about them. So that automatically makes them very interesting. What kind of gaps do we fill in this field called conservation genetics? We use genetics for species ID and trade monitoring, population structure, genetic diversity, what's called effective population size. We look at whether a population size has changed, reproduction, behavior, and more. Many, many, many different types of data can be acquired and used in conservation genetic projects. We work in my lab over the years on most of these topics, but I'm going to highlight a few today, sort of, telling this story. But first, I want to talk about this trade monitoring tool concept because I mentioned the finning. Why do we need genetic tools to monitor trade? Why can't we just look at it and say, oh, this is this or that is that? Well, we need forensics methods. The reason why we need to monitor trade is because we need to understand harvest or take. We need to know what species are being taken in what quantities from what populations in order to determine, is this population going down? Is this species in trouble? And we use that to inform extinction risk, which is that red list. But how do we usually identify sharks and rays? Is by morphological external characteristics, which are not present. If you just have fins, like I showed in that picture, you have to be able to identify from just a fin. Or if you just have meat, so meat sold at a fish market, just a fillet, what species is that? You can't tell. Not only that, even if you have a full animal, there's a lot of cryptic species. So cryptic species are those that look the same. You can look at them. You cannot tell the difference. You must have DNA to determine if they're separate species. Now, I have a student in my lab. His name is Brian Puerta Beltran. He is from Mexico City, and he joined my lab a couple years ago. And he's an amazing student. And he came to me, he had a project, he went out and got funding through a grant, just an amazing student. And I'm going to tell you a little bit about the work that he is doing on guitar fish. And he's in particular, he has been going to the fish markets in Mexico City since he was about 12 and collecting sharks and rays. He's just absolutely brilliant. And so we crafted this project together. So he's working on guitar fish, which are genus Pseudobatis. There are 9 species of this genus. And here we have again this red list graphic, and a lot are vulnerable. There's 1 endangered, 1 critically endangered, and 1 near threatened. But most of them are at some risk of extinction. This is their distribution. So there's 3 species in the West Atlantic, and there's 6 species in the East Pacific. Now, Mexico is very interesting because it has 8 of the 9 species. The only one that's not found is the Brazilian guitar fish. That's the one that's critically endangered, but it's not found in Mexico. All other 8 species are found in Mexican waters. We have no idea how they're being harvested, how they're being taken in order to determine the impact. So here we have, this is just a catch. We get information on these massive catches of these guitar fish weekly across the fish markets, but this isn't the only harvest. They're also sold as what is called pez diablo in Mexico. In other locations, they're called Jenny Henevers. And it is this tradition of taking these guitar fish and drying them and then modifying them to look like basilisks and all of these mythological creatures, like a devilfish, where it gets its name from. And so they cut it, they dry it, and they sell these to tourists, but they also sell them to local people because there's all this mythology behind they cure cancer, they cure anything. And so people will buy them for these cure-all supposed traits. So before January 2023, I'm going to explain why this date matters. These pez diablo were sold internationally. So you could buy these for a few pesos in Mexico. And on eBay, you could sell them for $200 to $300. Okay, so it doesn't take much to go, I can go and buy a couple, and then I can go sell them on the international market. Look at that markup. It was unregulated, which means there was no legal protection, and it was completely unreported. In February 2023, this changed. They listed this species under what's called Appendix 2 of CITES. What this just means is that internationally, in order to exchange animal products of this species, you had to have import and export permits that are very difficult to get. So I actually did do a search to see, and I could not find any on eBay for sale now. So that's good. But the domestic trade in Mexico is continuing. So what Brian wants to do, or is doing, is he is trying to document and quantify this take. So he's gone around to all these fish markets, bought all these pez diablos. He's taking morphological measurements, and he's also taking genetic samples because they're so kind of like cut up and twisted and dried. You can't identify the species from just the animal. He's tried to, but there's no confidence in that data. So we have to use genetics as well. So in addition to the fact that you can't really see many characteristics here to identify what this is, even if it was intact, they all look very similar. So he's sequencing ND2, the ND2 gene, to identify species. But it's not that easy. That would be way too easy. This group is taxonomically very complex. The question of what is a species hasn't really been fully addressed. So we can't trust GenBank. we can't trust that the information in GenBank is correct. So we're developing our own custom library, and we have got collaborators throughout the range of all 9 species, and they're collecting samples from throughout the range. We will then generate genetic data for them and then compare the Pez Diablo to this reference library. These are the IDs that Brian thinks he has based on morphology. So he's pretty sure about these. 8 are speckled guitarfish. This is vulnerable. So this is at risk of extinction. This one, he has no idea. And then he's got 21 that he's saying it could be freckled or it could be chola. These are both threatened with extinction. So, but what that tells us, based on these very preliminary data, is that we think that guitarfish that are at risk of extinction are actively being harvested and traded as Pez Diablo in Mexico, and this has not been taken into account. And so what that means is that they may be at a greater risk of extinction than we think. So when we finish these data, those data will go and be used in that next IUCN Red List assessment, and we might see those categories go up, that they are more threatened than we think. And there's this whole can of worms, this fact that we did with this morphologically similar group, there also appears to be phenotypic plasticity. So where you have one species that looks different in different locations depending on local environment. And so Brian is very excited about taxonomy. I don't really do taxonomy too much, but this is probably the first step into what will be his PhD into taxonomy of these Pseudobatis. Okay, so what I want to spend most of the time on is population structure behavior, and then I'm going to get into genetic diversity. Why do we need to define populations? Why does this matter? We need to define biologically appropriate management units. We might have a species that, maybe is in Chile, maybe it's also in Peru. Maybe Chile has its own legislation, Peru has different legislation. More than likely, those species don't get to the border and stop and say, I can't go any further. They travel, sharks and rays, many species travel very far. So if they're using waters of different countries, we need cooperation. And if they're not, we also need to know that so that we can understand the responsibility of countries. We also need to be able to say how many. Is the population increasing, decreasing? What is recruitment? What is the death rate? We have to answer all of these questions at a population unit. So conservation genetics has really focused on population genetics for the last 20 years. And then, of course, if we know the population, we assess the impact of threats. For example, with that Pez Diablo, we're just trying to document trade right now. The next step would be, what's the population impact of these things? Now, the most threatened families of all rhino rays. When I ask people this question, usually they say, when I ask, what is the most threatened shark? When I do outreach, I always get white shark. That's not true. Not true at all. It's just the most popular. The most threatened families are called the rhino rays. Has anyone heard of a rhino ray? I'm guessing no, right? But they're the most in trouble. The giant guitarfish, there's only 6 species described. There's probably more, but there's only 6 described. They are all on the verge of extinction. Wedgefish, 11 species. This one hasn't been assessed. 1 is okay. 9 are on the verge of extinction. We have no population genetic data for either of these, no species in either of these. We know nothing about these species. So that's not very helpful for conservation. Sawfish, there's 5 species, 4 are critically endangered, so on the verge of extinction, and 1 is a little bit better at endangered. I have been working on this group for almost 20 years, and we do know something about them. But we didn't start studying them until they became critically endangered. I started studying them in 2005, and they were assessed as critically endangered in 2007. So hopefully, in 5 years' time, we have some data for these. But I'm going to tell you about, I guess, the story of how we are advancing in this field. So sawfish, large, or pesieta, large benthic rays, so they hug the bottom, grow up to 7 meters in length. They're absolutely huge. Oh, I forgot I have a video. Is this going to play? Can we play this? Can you click on it? Will that work? Here we go. So that's what they use that saw for. They're using it. It is an electrosensory machine. They are detecting the heartbeat of fish in the water. And as you saw, they slash the fish, they pin it against the ground, and then they swim over it and feed on it. So that's why they have this saw. But the fact that they have this saw has made them incredibly prone to entanglement in nets. If you have this thing on your face and you are swimming around, they get caught and tangled in any kind of net that's in the water. They also have some of the biggest fins because they're 7 meters. So these guys were heavily, heavily exploited. One set of fins from a sawfish was $10,000 US. And this is where conservation genetics has a social science aspect, because what becomes very challenging is going to places where that $10,000 for one animal will feed their family for many years, maybe decades. How do you tell that person? No, you can't have that. It's that we have this social aspect. The saws as well. So when a fisher would catch one, they would chop the saw off. Now this sounds terrible. It is terrible. Back in the day, they thought it regrew. They thought it would come back. Now we know better. But fishing expeditions would go out. This was one fishing expedition in Florida. And this is what they came back with. They weren't even targeting sawfish. They just accidentally caught them. So they cut the saws off and they keep them as trophies, and they sell them. This is from an animal in Australia a few years ago. This is a fully protected animal. So this is still happening today. This also happened about 2 years ago in the US. One was cut off. That person was fined $5,000, I believe, and jail time. But you can still buy these today. So this one, I gave a seminar in September, I think, or October. So I had looked this up. So this was, look, it told me when it would arrive. It wouldn't, because it would be confiscated, but 1,000 Australian dollars. That's about 700 US dollars. So there's still money. There's still trade going on for this species. Oops, there we go. So let's go back to this. We've got narrow sawfish. This is endangered. And then we've got these 4 species. These are critically endangered. So these 4 are on the verge of extinction. In my lab, we work on all 4 of these species, different projects, different questions. I'm going to talk today about the large-tooth sawfish because we have the longest history working with this species. Critically endangered. They used to be what we call circumglobal. So you could find them, or circumtropical global. You could find them in tropical and temperate latitudes across all of the oceans. But because of bycatch, because of being directly targeted, this is their range contraction, about 61% over the last 100 years. The only place where they're found in what we call viable, in good numbers, is Northern Australia. Some of these other areas, you see them in blue, we don't know. That might be one animal that was seen in 50 years, but it takes a lot to say something is extinct. in a location. You need 30 years of data, for example. You need lots of data to say it's gone. We're very cautious like that. So this is going back in time, about 20 years ago. We knew nothing about the genetics of this species, nothing. And we wanted to know about gene flow. We wanted to know about genetic health. We wanted to know if there was inbreeding, if the population, I'll explain what this is in a little while, but if the population had decreased, we wanted to know what their population size was. And so how do you do this? This was 2005. This is where they were. This is where the population is. If you want to learn about them, you have to go there. So I started up here in Michigan. This is where I grew up, icy cold. And I moved to Perth, where there's Murdoch University. And I did my PhD there on sawfish genetics. And I remember the advisor at the time said, no, you can't do this. And I said, why not? And she said, you're never going to get the samples. You're never going to get the samples. It's this war. And I said, I'm going to get the samples. You just wait. Got funded by the Australian government. And yes, collecting tissue samples is extremely hard. It is in a critically endangered species. This is what Northern Australia looks like. It's this huge floodplain. It's inaccessible half of the year. The roads flood. This is a great road here. Whoops, wrong way. Giving it away. So here we've got just these dirt roads, and there's really charismatic creatures. So you don't go in the water with a saltwater crocodile unless you have a death wish. So yes, you could be in the field for months and come back with 3 samples. That doesn't work well. But after getting on the phone with people, emailing people, I managed to put together 150 samples from across their range in Australia, really just by networking and calling up various folks and say, hey, do you have any of these? Can you go get some? It took 8 years, but we got there. A lot of those were archived, sitting somewhere. So this is 2005. This is 20 years ago. We did not have the technology that we have today. So we developed genetic markers. We generated data for mitochondrial DNA. We went for control region. Why do we use control region? Because it's selectively neutral. If you want to look at population structure, you ideally want non-coding because you don't want selective bias in your data. So we did control region, and then we used selectively neutral microsatellites. Now this sounds really easy today, whew. But back then we had no genetic data for any species of ray at all. So getting primers to work without any reference library took some time, and it took a lot of trial and error. But again, we got there. So, we just had regular Sanger sequencing and just fragment analysis for our microsatellites. Now, why did we have both of these? Mitochondrial told us about female gene flow because it's inherited along the matriline. Microsatellites, biparental, right? So it's going to give you input from the mother and the father. What did we find? Okay, so you might be like, I don't know pop gen data. That's okay. We've got haplotypes here, and I've simplified this heavily. There were 2 common haplotypes in the mitochondrial DNA, haplotype 1. And as you can see, this was very common here in the Northeast, in the North, and it was less common here on the West Coast. Haplotype 2 was not very common in the Northeast or the North, and very common on the West Coast. And then I have these other colors. These just are haplotype, suites of haplotypes, so it's not one. It might be a couple haplotypes. And so these were unique to only one region. So what we have is a lot of haplotype 2 here, a little bit of haplotype 1, and then a whole bunch that were unique only to the Northwest. Here you only had, it was a very small sample size, so this is only 1 and 2. By here you had a lot of haplotype 1, a little bit of haplotype 2, and then again, a number that were unique to this area. And the East Coast was too small to do anything with, but it was interesting that nothing was shared. So what does this mean? Even if you don't understand population analyses too much, you can look and you can say this pie graph looks very different to this one, right? So we did FSTs, which range from 0, meaning one population, to 1, meaning very, very different. And we got an FST of 0.811, which was highly significant. Now, at the time, there were 6 other published population genetic papers on sharks and rays. And this number was insanely high compared to other sharks and rays, because we had this idea that sharks just roamed the oceans and panmictic huge populations. So this was absolutely unexpected to see this kind of number on these data. Then we did analyses on the biparentally inherited microsatellites, and we got the exact opposite pattern. We got, so these are just, don't worry too much about this. This is just from a structure plot where you're looking at, it's a Bayesian approach where you basically are looking at what is the likelihood of there being one population or 2 or 3 or 4 based on your allele frequencies. And the data said, without a doubt, there's only one population. And the FST here was 0.029. This is more what we expect to see for sharks, low in general, at the time, because we didn't know anything. What does this mean? Different results. So we've got lots of structure with mitochondrial maternally inherited markers, nothing with microsatellites, no structure with microsatellites. This hints at the fact that females are what we call philopatric. So this means that they're returning to the same location to give birth that they were born. So this is quite similar to salmon. And what's interesting about this particular species is it uses fresh water as nursery areas. So these adults are marine, and the females, we believed, were going back to their natal river and pupping. And the males weren't tied to that location. They didn't have to go back. And so the males are wide-ranging. And so this is what we call sex-biased dispersal or sex-biased structure, where the males are wide-ranging and the females are philopatric. Now, at the time, again, this is a while ago now, this is about 2011, 2012 now, this wasn't observed in any species of ray. So that was really cool to see that as a first for a ray. And we hypothesized that, so we made this hypothesis of philopatry to rivers, but we didn't have the sample sizes to test individual rivers. So we learned a lot of information, which was used in management, but we had questions. Were females, this was 2012 now, were females philopatric to rivers? And if males are moving widely, how wide? Are they going into other countries? We need to know that. The Australian government desperately wants to know that. The first part of the question was answered in 2015. So it's kind of funny because I was offered this job as a postdoc in Australia, and I turned it down, and I went back to the US instead because they just wanted me to continue this work, but I was like, I need to diversify, I need to work on other species, I need to do other things. But this paper came out those few years later, which was really exciting, and they demonstrated that hypothesis was correct. They used whole mitogenomes because by this time technology was advancing. And again, don't worry about too much about the details here, but what you can see is that there's clusters of colors together in different rivers. So this is in the northwest of Australia, and they're all different. This one is in northern Australia, and they're very, very distinct. And so what this demonstrated was that, yes, different rivers were different maternal populations. So this was really exciting to see. And a first in sharks. The next question, this is what the Australian government still wants to know, is there connectivity from elsewhere? Especially they're interested in Southeast Asia, because Southeast Asia doesn't have the same protections and are still harvesting sawfish. So Australia wants to know if their sawfish are moving to Southeast Asia and potentially being taken. So interestingly, we are working on this in the US for Australia. But how do you do this? How do you go collect samples? There's none left, right? There's very few left. How do you do this? Remember those old saws? We are collecting genetic samples from these old saws to answer this question. So we go into museums and private collections, collect some tissue, and we have a little hashtag called Saw Search. And we've traveled all over the US. These are two students. This is Kelsey Smith. She was a PhD student. And this is Anne-Marie Fearing, who's also a PhD student. We traveled throughout the US. We traveled to the UK because the UK has some of the oldest, largest collections. And they have so many, not because sawfish were over there, but because of the old trade routes. So they have massive collections. I sent a student to Australia. I also went to Australia and got some more. And then we have a network of 24 researchers who all collect samples for us. Very collaborative. We bring it back in the lab after a stack of paperwork for import like this. And this is what the DNA extracts look like. And this is probably very different than the DNA you look at because yours is nice and clear. Ours is disgusting. And it doesn't look like this, right? It is old and it was never preserved for genetic analysis. It has been degrading over time in sometimes horrific conditions. So we only get teeny tiny fragments of DNA. So I put a gel in here just so you could kind of see, like, you know, the difference between this is what we have to work with. It's the same for eDNA, which I'll talk about tomorrow and Wednesday. But it's just terrible DNA. It's awful. It's like, what do you do with that? So we are able to sequence it. This is sequence from that terrible. There's different ways that you have to process these samples. You have to sometimes pre-process them in case they have chemicals in them, paint, but we get sequences. So I'm going to share some of our preliminary data. And I've taken a smaller fragment here to communicate something more simply. And then I'm going to show you the next slide with a larger data set. So this is from a very tiny fragment. This is still the control region. And we want to compare our data to contemporary data for another component. So what this is at the level of each ocean. And it's the same concept, different colors, different haplotypes, or different genetic sequences. And this is the Indo-West Pacific. This is the genetic composition of these. West Africa, where we have 43. West Atlantic, where we have 20 in this particular data set, and here where we have 11 in the Eastern Pacific. And again, if you look at this, look at the colors, look at these, they're very, very different, extremely different. When we, on our bigger data set as well, when we do our population structure analyses, this is the division. So the Eastern Pacific population is distinct from the Western Atlantic, which is distinct from the Eastern Atlantic, which is distinct from the Indo-West Pacific. Now, if we do the whole control region, and this is really hard work because we can only get tiny fragments that we have to glue all back together, basically. We see that they're very different here as well. The green is the Indo-West Pacific, and these haplotypes are the Eastern Atlantic, Western Atlantic, Eastern Atlantic. But this is really interesting to us that the Indo-West Pacific is very distinct from these other regions. We do an FST, again, a very large FST of 0.726. So these are highly, highly structured animals. And that we think there's a lot of structure along the matrix line within regions, especially within the Indo-West Pacific. So we're working now on answering this question for Australia, where we suspect that we have structure within this region, but we need larger sample sizes. We have them now. We got them through a collaborator who collected 40 samples from Papua New Guinea. Took 1 year to get them exported into Australia. And now we're working on the export from Australia to the US. So we're working on that right now. And this is Anne-Marie Fearing. She actually has a Fulbright to go to Brazil in February. And she is going to, she's working on developing 40 to 50 microsatellite loci to complement the mitochondrial sequence data. That's going to allow to assess male bias dispersal and another component that I'm about to talk about. But you might be going, why don't you just use SNPs? Why don't you just use SNPs? Oh, somebody tried. So at the time when I was applying for these grants, I was very, very early career, and I felt like I couldn't take high risks. But a colleague of mine who's very well established took that high risk for me ahead of time. And they tried to get SNPs from shark fins, and it was an epic failure. The reason is because the DNA is so bad. We need technology to get better for low copy DNA. It's just not there yet. So we're going to do this approach in the meantime. So this connects to talking about genetic diversity. This is something that I find extremely interesting. When you have a huge decline in abundance, we usually see population bottlenecks. So if you have a very large population, so here I've got some gummy sawfish, different colors, means lots of diversity. And then if there's a sudden decline where you wipe out a large percent of the population, only a subset of that genetic diversity survives. If the population grows, it has lower diversity than the pre-bottleneck population. Why does this matter? Inbreeding depression. So that's where you've got, you don't have a lot of polymorphisms, and you end up with a small gene pool, and you end up with relatives, and you can have this concept of these deleterious recessive alleles where relatives are more likely to carry recessive deleterious alleles. Relative and relative get together. It's not good. Susceptibility to disease. The Tasmanian devil has unfortunately been the poster child for this. The Tasmanian devil is subject to this facial tumor disease, and it was almost completely wiped out from the island of Tasmania. There was one population that harbored resistance. And thanks to that population and that resistance, there's hope for Tasmanian devils, because now they're trying to confer that resistance through breeding, but also, through genetic approaches into other populations. So they're working on identifying that. And ability to adapt. So the whole basis of adaptation is going to be genetic variability. If you're all genetically the same, there's the potential to have similar responses. Now, the problem, especially in sharks and rays, if you have a population size, bottleneck, and then over time it recovers. It looks great on the surface, and the conservationists get all excited about this. And then I'm usually the doomsdayer who's like, oh, because you got to look at the genetics of it as well, because that might have plummeted, and that's not going to recover, especially sharks and rays, because sharks and rays have got the slowest mutation rate known of the vertebrates. They have their mutation rate is exceptionally slow. So for them to recover any diversity is extremely challenging and unlikely on scales relevant to management, which are medium to short. Genetic diversity, there's not a lot of attention on this for sharks and rays. When do they consider it? I always get into arguments with people about this. They care about it for ex situ, captive breeding. Then all of a sudden they want the genetics of everything, and then they want to know if this is related to that. But that's really it. It's not considered in those extinction risk assessments that I showed you. And there was research that came out last year, I believe it was, where they actually looked at it and they noticed for species on the verge of extinction, they probably face a higher risk of extinction than we think because of the genetic aspect, which has been ignored in those assessments. So sawfish, perfect example of a crash, a population crash that's moderately well documented. Genetic diversity data that we have for sawfish, yes, we have some so remember there's 5 species, we have it for 4 species. These are sample sizes. This is an index, this is haplotype diversity. It ranges from 0, where all your individuals have the same haplotype, to 1, where every individual is different. And you can see these are middle of the range, right? Average allelic richness, 10 to 14 alleles. Average observed heterozygosity. So this is going to be, your heterozygotes that have different alleles at a locus. This also ranges from 0 to 1, 1 being good, 0 being bad, and they're higher. But what does this mean? These are just numbers. What does this mean? What are healthy levels of diversity for sharks and rays? We don't know. We have no idea. And one reason we have no idea is because there are no pristine populations left. They've all been in decline. So, is this reduced or is this okay? Does it matter? And how should we manage populations in light of diversity? Because with that Tasmanian devil example, if you would not have preserved that population that had resistance, they probably would be on their way out. So we're using these old saws again, this DNA. We're looking at spatial declines associated with range declines, and population structure is very important for this. But we're also looking at temporal declines. So we're looking at what was diversity now to 50 years ago, to 100 years ago, and 150 years ago in Australia, Brazil, and Papua New Guinea, because those are the locations that we have sample sizes for, and these may be promising. So these are just preliminary data. But remember I showed you that range contraction of 61%. The genetic data suggests a loss of 80% of the genetic variability associated with these range declines, which basically restricted populations to Australia. So, and again, this is just a subset of the data, but you can see out of 57, 23 different haplotypes is quite high for sharks. That's very high for sharks and rays from the historic populations. And here's our Australia diversity right here, but that's a very small subset of what we had globally. If we look just in Australia, because of course Australia is interested in their self, they want to know, well, how are we doing? Hoping that I'll tell them they're doing better. And I do get to tell them that. So their diversity has been stable for at least 60 years. So the frequency and the types of haplotypes we're seeing are very similar. So they're doing a good job. So because it's viable, so far we've done it, we're going back to 100 years and 150 years, they're pretty okay. We're not too worried about losing this species globally at the moment because we have this Australia stronghold. So the genetic diversity in Australia, this is what we would say is secure from a conservation standpoint. All of this is very insecure. This could all be lost, and we don't even know what's contained here, which is what's the scary thing. So identifying and securing viable populations outside of the Indo-West Pacific is a very high conservation priority based on this, based on the fact that this is genetically very different. There's very high levels of structure, and it is a distinct genetic lineage. So we're really worried about it. So maybe these areas, maybe, we don't know. We're exploring this, which I'll talk about on Wednesday, showing how these things connect. So what happened, with this species? We've been studying it for almost 20 years. Started in 2005. It was critically endangered in 2006. We got it listed on CITES Appendix 2 in 2007. This means that international trade stopped, except for aquarias. Sawfish were still being taken for $10,000 each from Australia and sold internationally. When I started this work in 2005, despite the fact that they were critically endangered, they were not protected. So we worked to get them protected in state and federal waters, and they had complete protection in 2010. In 2012, we finally got it uplisted to Appendix 1. So I got to go to Rome for this to make the case that the aquaria trade should stop, and we got that stopped. They were listed on the U.S. Endangered Species Act in 2014. And you might say, well, they were historically found in the U.S., but they're locally extinct there now. Why protect them? It's really a mechanism to prevent trade, international and national trade. And we just use these in the new red list assessments that I showed you the pie graphs of this year. I just want to briefly talk, this is going to be really quick, but I wanted to show you where we're going in this field, because this links up to what some of you do. Conservation genetics to date has focused on non-coding, selectively neutral markers to describe populations. We're finally moving forward. We're moving into larger data sets and developing novel tools. Part of this is because costs are decreasing. There's not a lot of money for conservation, and we have to make very good use of it. RNA transcriptomes have a lot of potential here. You can generate them for relatively cheaply now, and the use you can get out of them is absolutely incredible, and we're still exploring this. So we have we're working on the small tooth sawfish for this. It's also critically endangered. This one was only in the Atlantic. And you can see, so orange is extant by country, red is possibly extinct, gray means we don't know. This is really the only place you can find this species globally. So this species has undergone the largest decline out of all sawfish. In the US, their conservation priorities: number 1, minimize human interaction. Number 2, protect and restore habitat. And number 3, ensure abundance increases. How does an RNA transcriptome help with this? It does. Number 1 and number 3. So this was my job to go to the US government and say, yes, we need this. And I convinced them to give us money because they were like, how is this going to help us? So we got tissue samples. They had to be opportunistic. You can't go out and sacrifice sawfish. So we managed to get a dead female who was fresh in 2018, and then we collected blood in 2019. We have a RNA transcriptome. We have a low coverage genome for males, and the Vertebrate Genome Project has a genome for females. We put all these data together. I am not a bioinformatics person. I am not a developmental biologist. I am a conservation geneticist who knows just enough to work with folks who work in developmental biology, bioinformatics, and we come together. So we have this paper, it's in BioRx. It's in the final stages of being published. Alex is the bioinformaticist on this. Taya was a grad student. So this basically shows that we did much better with an RNA transcriptome, using that to annotate the genome, because it's this that combination of things has produced the most complete gene set for any shark or ray to date. And we're using this data because it's funded by the US government. We have to use these data to support conservation. So what we've been doing, so again, I said to date, mostly we focused on selectively neutral things. Now we're moving into things under selection, coding, these kinds of loci. So I needed to produce to the government, why did we just give you money for this? What are we doing with this? And so we identified genes that relate to their priorities. And that's the key in conservation. You might think something is cool, but if the conservation managers and government don't want it, you don't usually end up pursuing it. So we have genes involved in electroreception, male-specific genes, MHC loci, and hormones. So I'm just going to talk very briefly, like one slide each on some of these things. So I said the #1 goal of the US government for conservation is reduce and minimize interactions with fisheries. So we want to understand the mechanics at a molecular level of electroreception, because remember, they've got that saw in their face and they're so different from anything else. So we aligned this data with other species, and sawfish are really cool with their electroreception. So sharks and rays have different types of electroreception machinery. And what they have here is they have this, this calcium potassium channel. You have an influx of calcium, an efflux of potassium, and then you have this depolarization, which elicits a neurotransmission. So this differs between sharks and rays. These things have a combination of the two. They have different electroreception machinery. And usually there's this inhibitor. Sawfish don't have this. Every other elastomeric looked at has this. So we think the fact that they're lacking this inhibitor is why they have such heightened electrosensing. It is exceptional. So what are we using this for? There's work being done on deterrence, developing deterrence. And we've got kind of two different groups working on this. This is the Australian group. Interestingly, this is my significant other. But they're working on electrical fields. So they're stimulating sawfish with electrofields, but just looking at behavior. What does it do? And so our side, the molecular side, is saying, let's not focus on what they're doing, because this paper found they have a response, but it's not strong enough. And we're saying, let's learn about what's happening at a cellular molecular level in order to guide deterrent behavior. So, because nobody knows that. To me, this is just throwing things at a species and seeing what they do instead of actually understanding it. So the next step in this is you know, we might, the permitting could be tricky. We might have to do some cloning and things, but basically do these experiments where we are looking at molecular and cellular level responses to stimuli. So the government is extremely interested in this. And then we'll combine it with the behavior people. The other thing that's been really interesting with this data set is MHC complex. There's no MHC data for sharks and rays. It's another black box. And so we're really interested. There's a colleague I have at the university, at the Field Museum in Chicago, because there's so much potential. We can look at adaptive potential. I was talking about genetic diversity, but we've only looked at it at neutral loci. But what about this? You know, immune response, mate choice, sexual selection, inbreeding avoidance. These are all things that can be studied through MHC markers. So we haven't taken this very far. What we did was we identified the major classes 1 and 2, got some loci, and just saw if they were even polymorphic as a starting point of what locus to use. And this one looks really promising. So now we're following up with this to look at mate selection, because there is a lot of evidence that says that, now how they do this, I don't know, but they choose a mate that's different from them at their MHC locus in order to, you know, avoid inbreeding and enhance health. So this is one of the lines where we've got all these lines of research stemming from this transcriptome work. And then we've got these male markers. I've been interested for a long time to develop a genetic sexing tool because I have all these sawfish saws without sex, and I want to know their sex. This has never been done in sharks and rays before. It's common in other taxa. So we want to do that. Male specific SNPs, which we've got, and basically learn about male biology, because until this, we didn't even know if they were XY. So this combination of male and female genomes and the RNA transcriptome, like we know this is just going to host a lot of new projects. You know, we've been talking about how we can harness this transcriptome in even additional ways, which is really exciting. Hurdles exist. Funding, lack of expertise, which is why you need good collaborations, and really uncertainty in how else we can use these data. So these are all the folks that I need to thank for funding the conservation genetic work we do. My graduate students, my postdoc, my undergrad students, all of my lab alumni. When you do conservation, you work with a lot of people. These are all of our collaborators and all of the museums that we've visited. So it takes an army if you're going to work in conservation on these species. So I'll take any questions. I think I went way longer than I was supposed to, but. How's my Spanish?
Oh, this is. Oh, yeah. How was the process to get the museum samples?
The which samples?
From the museum
Oh,
samples.
the museum samples?
In terms of laws, or I don't know if the samples were from different countries or.
Yes. Let me go back to that. I kind of skipped over that. So this is one of those things that, you know, I'm sure all of you are learning that one slide is 2 years of work in some instances. So we actually had to do a separate pilot. I did a pilot study when I was still in school to figure out how to do this step. And it took a full year to 2 years to figure this out. These are in awful conditions. You don't want the skin, because that top layer of skin is contaminated with DNA from everything else, especially because the saws are stacked up on top of each other. So we have to take the top layer off. And then we take this sort of, you see this under tissue here? This white flaky tissue. And that's what we're actually getting our DNA out of. We just use a D&Easy blood and tissue kit for this, but we have to use like double the amount of tissue. If there's been any chemical on it, we have to clean it first with a PBS buffer to get rid of that. But yes, it took a year to figure this out because I was trying to extract DNA from the teeth. You'd think the teeth would be good, but they're terrible. And inside, and this actually gives reliable DNA in comparison to everything else. So actually that was my first publication back in the day, was comparing ways to reliably collect, extract, and sequence this DNA. You have to target tiny, tiny fragments and then put them together. Once in a while, a museum, I feel like we could do this to grade museum storage quality, because sometimes we go to a museum and the DNA is beautiful. So we do have some where the DNA is good enough and we're doing mitogenomes with those, the whole mitochondrial genome. But by and large, the average sample is very poor quality DNA. But yes, we get to travel all over.
Thanks.
The museums are fun. You never know what you're going to find down there. Like you
Yeah.
pull out a white shark head or a colossal squid. They're fun.
Question, guys? Perfect. So, thank you very much, Nicole. It's a very interesting talk. We are now, I think that we don't have many expertise in ecological aspect.
Mhm.
Of course, in the biochemistry, that these guys are all biochemistry.
Yeah.
So their principal interest maybe is physiology.
Mhm.
But the idea here is the great opportunity that there are to genomics in conservation.
Yes.
It's not just in human or goat or Arabidopsis model organisms. There are a lot of opportunities for genomics.
And a lack of expertise, right? So like most people who are trained as conservationists are getting ecology. So we don't know about
Yeah.
the other side. So there's so much opportunity there.
So like Nicole say that the people from ecology field here, from the genomics here,
Mhm.
and the idea in the ideal world, of course, is the good job, me. So because this is very important. So there are there are many options to go to the goal with a Fulbright fellowship. Just make a stay there. So that's very important.
Yeah, so the transcriptome stuff I think I mentioned. Someone who's not an ecologist, not a conservationist, not a shark biologist is the person I worked with on this one. He's a developmental biologist. He mostly works with early chordates. So, this person, bioinformatics, developmental biology, bioinformatics, cell biologist, ecologist, these four in the middle.
That is very important. More question? No. Thank you.
Okay,
Nicole.
you're welcome.
It's a great talk. Thank you very much.
Oh, yeah.